And we want a number of contigs that are over 10 and possibly over 100 kb. We want to look at N50 over N90, which are basically weighted means of the data. And then we want to know, we want to have the biggest assembly we can get. So I just wanted to compare these two because I used Meta in my PhD, and then Meta came along around 2012, 2014. And just to compare these two assemblers, so I took some small IDE data, and I assembled it, and I show right off the bat that I take the same data, treat it the same way. I get longer pieces and bigger contigs just by using a different algorithm. It's insane, right? Take the same data, assemble it with a new thing, get better data out. Pretty cool. The biggest metagenomic assembly that I did in my PhD was 30 gigs. It's the most data I ever got. When I walked into Janet's office, she gave me one sample that was 250 gigs. It took me 2 years to figure out how to get these done correctly in our cluster. Once again, I have a study here, which is from some dirt from England, and we have airflow samples. So you can take the same data. It takes upwards of months or many weeks to get an assembly, or I can do it in a couple of days using this assembly. That's why it's so important. And the most highly important, the biggest data, 279 gigs, we've got 6,000 over 10K, a maximum over 100 gigs, which is unheard of in soil, and 32% of those reads are actually mapped. So we can bin from that data. As I mentioned, I didn't talk about it in great detail in the beginning, but I helped develop this new technology, and this is of course published in Systems earlier this year. And the idea is this, and it's kind of complicated. So how do you take a short read sequencer and make it a long read sequencer? So what we did is we developed molecular technology, and so you take very, very long pieces, about 10 kb. You then put Illumina adapters on them. You do a serial dilution down to where you have 200 copies, and you put that in a 384 well plate. You do a long PCR to then clonally amplify. So you have low diversity of 200 molecules, so you have, we'll say, maximum. You have 200 organisms, right? And so you clonally amplify those 200 organisms. So you limit the diversity of the sample. And then from there, as I go too fast, you do some fragmentation, you then do a PCR in adapters, and then you sequence. And so the idea is that each individual well will have its own unique assembly. So you can go from 10K down to 500 base pairs, do 100 base pair reads, and then reconstruct your 10K or 8K piece. So you're able to do a short read sequence